Very good afternoon to all the participants. I'd like to extend a heartiest welcome to all the participants who are present with us today for this session. We at Sympathy Institute of Homeopathic Pharmacy and Hospital believe to spread and elevate the knowledge of homeopathy for all homeopaths. Today, I am your host. I'm Dr. Aisha Shamsi, Principal of Sympathy Institute of Homeopathic Pharmacy and Hospital, and I'll be hosting this session. And for this today's session, we have a very important and crucial topic, that is standardization of homeopathic drugs and potentized medicine. Because standardization is the very base from where all our drugs start. So it is very important to know about this subject. Today's session will be presented to us by Dr. Shreya Deshpande. She has been a dedicated scholar and she's currently serving at Hmasa Homeopathic Medical College. She's a scholar and a senior in the Department of Pharmacy. So during this session, we'll be having a presentation session, which will be followed by question and answer. So any participant who are having the message so that Dr. Shreya can answer at the end of this session. Over to you, Dr. Shreya. Thank you for being with us today and spreading this knowledge with us.
My sincere thanks to Sympathy Institute of Homeopathic Pharmacy. Is there any disturbance in my voice or screen share?
Yes.
Am I audible now, ma'am?
Yes, you are very much audible. Kindly continue.
Okay. Thank you. A hearty welcome, one and all. My sincere thanks to Sympathy Institute of Homeopathic Pharmacy, the first and foremost homeopathic pharmaceutical institute and also hospital. Also, my thanks to Hmasa Homeopathic Medical College, our directors, all the faculty for your support. Without any delays, we'll dive into the topic, standardization of homeopathic drugs and potentized medicines. Firstly, why is standardization in homeopathy important? So whenever any patient takes any medicine, they expect our medicine to be genuine, that are safe and consistent. So standardization today, nothing but quality control of homeopathic medicines. Standardization of homeopathic drugs refers to the establishment of defined quality parameters to ensure that all the medicines are authentic, pure, safe, uniform, and they are reproducible throughout their manufacture and use. That is, from the start to the end, all the medicines, how they are, including the raw material, how they are collected, processed, manufactured, and distributed, that can be known through standardization. It is a process of having a product, any medicinal product or any quality, in uniform standard as mentioned in our components. So the main aim of standardization includes quality control and assurance. Quality assurance, giving guidance and giving genuine quality. Coming to quality control, how the testing, inspection, consistency, purity, and process monitoring is done is covered under quality control system. And quality assurance, how we are assuring, how you are giving them the confidence that your medicine is safe and effective to use. It includes safety and compliance, documentation, and continuous improvement when there are any discrepancies found. Quality control. It includes quality control system with various tests to perform at different stages of manufacture. As I said, from start to end, that is from raw materials to finished products. Next, quality assurance and safety. You are giving them a confidence, you are giving them a promise that your medicine, our medicine are safe to use effectively. It also involves an assurance and assessment of quality of products with reference to pharmacopoeia monograph. This is the reference. Pharmacopoeia is major the reference for our medicine that are prepared. And the objective of standardization is to ensure that every batch of medicine possesses the same quality, identity, strength, and pharmaceutical characteristics as mentioned in the official pharmacopoeia standards. Main primary focus includes identity, purity, safety, what are the quality and stability, and the strength and potency of the medicines. Identity and purity, how the raw materials are identified, how the drugs are prepared, verifying that the correct drug source material is used, ensuring absence of contaminants and adulterants. No any sort of contamination or raw material adulteration are done. That is to be ensured identity and purity of the raw material collected. Coming to safety, toxicological and microbial risks. Next is strength and potency. Maintaining vehicle quality, most of
Dr. Shreya, I think you are disconnected. I'm making a co-host.
Okay. Sorry for the disconnection, guys. Is my screen audible, visible now, ma'am?
Yeah, very much visible.
Good, ma'am.
Please continue.
Yeah, continuing this. The main primary focus for standardization, as said, identity and purity, safety, strength and potency, quality and stability. We have seen identity, purity, and the safety, reducing the toxicological and microbial risks. And now strength and purity, maintaining the vehicle quality. Majority will be using alcohol as the vehicle. Next, purity and consistency should be maintained in our preparations. That is the primary motto. Next is the quality and stability, estimating active constituents present in crude drugs, whether alkaloids, glycosides, any sort of active constituents present in crude drugs, and ensuring stable therapeutic preparations. Here, stability includes the capacity of a particular formulation to remain within its physical, chemical, microbiological, therapeutic, and toxicological specifications for a prescribed period, as described in pharmacopoeias. And what is the need for standardization? Coming to preparation of drugs, raw materials can be collected from various sources. Here, raw materials vary according to geographical source, climate, their harvesting season, and storage conditions. You need to check whether the medicine prepared in Telangana, in Delhi, or in Germany are the same, are the three formulations. Suppose taking Arnica mother tincture, I have prepared in my college, it is prepared in Delhi, it is prepared in Germany. We need to test whether are all the three same, because they have been grown in varied areas and the climate is varying there, and storage and transport conditions also matter. So here standardization plays a vital role. How the materials are collected, how the preparation of medicine is being done. For this, standardization plays a important role in this. Next, homeopathic medicines undergo serial dilution and succussion, requiring precise manufacturing procedures. Mother tinctures or dilutions or tablet triturates, all these require different procedures for preparing. So after preparation, are all them same? Going to higher dilutions, the presence of raw material may vary. You need to find out the percentage of our material, raw material used in, suppose you are taking 50 millismol potency. How much is the percentage, or how the medicine is present in it or not? You can prove. There are many advanced techniques we'll be seeing in the coming slides. The presence of medicine is known through standardization with these advanced techniques. Next, contamination during transport, collection, processing, storage, or while dispensing. Whether any adulteration has happened or not, you need to test, and standardization and methods of standardization help in that. Regulatory authorities require uniform standards for quality assurance. Any regulatory authority, you are selling the homeopathic medicine, you are buying them. For that, you need authentic formulas that are mentioned in the pharmacopoeias. So you need to make sure whether your prepared medicines are following pharmacopoeial standards or not. The acceptance for a medicine comes with after it follows the pharmacopoeia rules as mentioned for each drug in that particular monograph. International trade and pharmacopoeial harmonization demand reproducible products. Reproducible products, nothing but you have prepared dilutions for serial dilutions, and international trade, suo moto, giving and taking medicine from other countries, foreign trade, that all is accepted after the standards of pharmacopoeia of that particular drug are met. So that is a need we have seen. Just a glimpse of sources of homeopathic drugs. Plant kingdom, animal kingdom, mineral kingdom, nosodes, sarcodes, isodes, allersodes are all sources of homeopathic drugs. Each source has a particular preparation that is different for each kingdom. And now we'll go with the methods of standardization. There are 5 methods that are organoleptic evaluation, microscopic, physical, chemical, and biological evaluation. We'll see one by one. Coming to organoleptic evaluation, it nothing but qualitative evaluation. Just by seeing the physical nature or external appearance, we'll be able to say how identify the medicines. Like organoleptic evaluation refers to qualitative evaluation of drugs by organs of senses: eyes to see, nose, smell, tongue to taste, and skin to feel, or by touch. That includes organoleptic evaluation, shows the macroscopic appearance, its odor, taste, and the feel of the drug by touch. It includes external morphology, shape and size. Various shapes are there by you. We need to identify by seeing them. So this organoleptic evaluation, it will not dive into quantitative, just by qualitative appearance we'll be knowing. External morphology, that various shapes are there as given: aconite, conical in shape, and sarsaparilla, cylindrical, and all the sizes based on the length, breadth, and diameter as measured in centimeters or millimeters. And next are the external color. They are also varying. As seen, aconite and sarsaparilla are given. Color varies, purple and reddish. Varies from white to yellow, gray, yellowish, brown, reddish, orange, blackish, brown. Many colors are there. That shows the identity and uniqueness of the plant or the medicinal product. External markings on the leaves or any organs. They may be furrows, parallel, wrinkles, ridges and valleys, or any transverse markings of various types as shown. And fractures, how when subjected to pressure, how a plant or plant part breaks. When the plant is budding, the break is from inside out, and when it is breaking, it is from outside to inside, center to periphery. That also matters. Odor, different types of smell like aromatic, camphor smell, turpentine smell, ethereal smell, etc. Each product or each plant or medicinal product has different odor, and tastes are also different. They may be sour as in acids, salty for saline products, sweet as in saccharin, alkaline, bitter. Some may be tasteless, insipid taste, astringent, pungent, starchy taste as in carbohydrates, etc. That is regarding organoleptic. And microscopic evaluation, it helps in identifying organized drugs and searching adulterants in powdered drugs. Through microscopic evaluation, we'll know the arrangement and individual tissue character of plants and their parts. Histology, the basic stomata or the vascular bundles or the inside of the plant parts, that is the histology, can be known through microscopic evaluation. Details of histology with microscopic appearance of drug in section view and powdered form. Why here we are mentioning powdered form is, as randomly a medicine is given and you need to test it in the laboratory. All these tests will be helpful to know in the powdered form. That will be helpful in identifying the drug given to you, or you are going to test the drug. Microscopic evaluation also includes microchemistry, isolation of drug constituents by microsublimation, microextraction, filtration, crystallization, identification of constituents of drugs by crystallography, melting point determination, physical test, chemical test, and some color reaction tests. That is regarding microscopic evaluation. As seen in the diagram figure of Aconitum napellus, we can see the vascular bundle of the root and the stomata of leaf, or the root powders and vascular bundles of the stem. Coming to physical evaluation, we'll be knowing the physical constants like solubility or the specific gravity, density, viscosity, melting point, and refractive index. These physical constants help in identifying the external qualitative or the quality of a product given to us for the testing. Specific gravity of a product will be knowing compared to specific gravity of water. And the same way, density, viscosity, how the melting point is varying. This helps in adulteration if any sort of decompositions or any improper preparations have happened, or if they are suspected in any given drug sample. The refractive index, how the light bends through the liquid or the mother tincture or dilution you are going to get tested through that. So these are the physical constants we need to test in the physical evaluation. And some advanced techniques like chromatography, spectroscopy, and fluorescence tests are included in physical evaluation. Through chromatography tests, we are going to identify how the mixture, suppose any mixture of a compound or a mother tincture is given, you need to separate the chemicals in that. Chromatography helps in the separation. It helps through identity, purity, marker compounds, adulteration, and also maintaining the consistency. Through TLC or thin layer chromatography, a sample is being tested. It may be done manually. And the difference for TLC and high performance thin layer chromatography is in HPTLC, multiple samples are being tested. As I said earlier, arnica mother tincture, you need to find out the prepared mother tincture from 3 different places. You need to sort them out. So when you are testing in HPTLC, as shown in the figure, 3 components are there. You will place them as spots, and the result can be shown through retention factor value. That is the distance traveled by the component by distance traveled by the solvent. Solvent, nothing but there is a, for thin layer chromatography or chromatographic studies, particular solvents are used like silica gel and all. So you need to test them. If all the 3 follow the same and the same path, then it shows there is no sort of adulteration that has happened. If there is any difference in the 3 spots, there may be kind of adulteration, or the raw material may be different of the prepared mother tincture. That mixture of compounds can be studied through chromatography. It acts like fingerprinting. For each person, there is different fingerprint pattern. So in the same way, for each drug, there is different chromatographic pattern. Chromatography, nothing but chroma is color and graphy is writing. So color writing, for each product or for each compound, there is different and it is unique for that product. It should be tested comparing to other products. Next is the spectroscopy. UV visible spectroscopy, infrared, and NMR, nuclear magnetic resonance spectroscopy. Spectroscopy shows the molecular structure of the drug and the functional groups present in them, also the chemical identity of that drug. Every molecule interacts with the light differently. The principle of spectroscopy is nothing but interaction with UV visible light, that is UV spectroscopy, and IR, infrared radiation, also NMR, nuclear magnetic resonance. The molecular structure and the presence of functional groups, whether acids, ethers, this is useful in animal and mineral kingdoms on the powdered form of drugs. Next is the fluorescence test. It shows how different compounds glow under UV light. When compounds are exposed to ultraviolet light, different products like aconite, the physical phytochemical present in it, aconitin shows light blue color, and berberin from Berberis vulgaris showed yellow color, and emetin from ipecac showed orange color. The reaction of certain drugs, either in powdered form or in the sectioned surfaces with filtered UV light, helps in determining the adulterants. That any difference in the color shows the presence of any adulteration that has happened in the given products. Many alkaloids show distinctive colors under UV light. As seen the examples, aconite light blue, berberin from Berberis vulgaris shows yellow, and emetin from ipecac shows orange. Next, that is regarding physical evaluation, showing the physical constants. Coming to chemical evaluation, it covers isolation, identification, purification, and characteristic determination of drugs having active principles. In short, we are studying the chemical constituents or the alkaloids, flavonoids, and the phytochemicals present in the drugs through chemical evaluation. It helps in detection of alkaloids, carbohydrates and glycosides, fixed oils and fats, volatile oils, saponins, proteins and free amino acids, gums and mucilages, phenolic compounds and tannins. Analytic chemistry may be qualitative or quantitative, as seen in chemical. Some chemical assays include color reaction test, acid value, iodine value, saponification value, and ester values. Chemical evaluation, mainly it shows the presence of active chemical constituents and the medicinal action of the why chemical evaluation is needed. Medicinal action depends upon the chemical constituents. The presence of phytochemicals in plants and the chemical constituents in the animal or mineral kingdom drugs, the presence of constituents in them plays an important role in the action of the remedy. Through chemical evaluation, presence of alkaloids, glycosides, tannins, volatile oils, proteins can be known. They help in confirmation of the purity and quality of the drugs. Next, coming to biological evaluation. This is done only when chemical evaluation is not suited or when it is not sufficient. Through biological evaluation, testing biological activity. Nothing but, as we said, in chemical, the presence of chemical constituents or phytochemicals is known. If it is not known, we'll be going to biological evaluation that includes pharmacological testing, bioassays, toxicological evaluation, and microbial testing. Testing biological activity in the living systems, it shows actual pharmacological activity. Pharmacological testing, bioassays, toxicological evaluation, and microbial testing. The assays on living organisms and on their intact or cut off organs are indicated. Second. The assays on living animals and on their intact or cut off organs can indicate the strength of the drug or their preparation. As living organisms are used in this, these assays are known as biological or bioassays. As said, the testing is done on living organisms. For example, cardiac glycosides present in drugs like digitalis, they are bioassayed on cat, frog, or pigeons, on living organisms. This shows animal toxicology and animal proving. Evaluation of ergot is done on rabbits' intestines, and testing arnica mother tinctures for wound healing in animal models also plays as an example for biological evaluation. Coming to standardization of mother tinctures, we know mother tinctures are the foundation or the main base of homeopathic preparations. They can be subjected to both qualitative and quantitative analysis. Parameters evaluated in mother tincture analysis include identity tests to confirm the source material, as we have seen in earlier tests, and alcohol content determination. Major part of mother tinctures include alcohol as a vehicle. So confirmation of the source material and the presence of vehicle used, like alcohol content determination, it is measured because ethanol, the ethyl alcohol acts as a solvent, preservative, and extraction medium. Also, we'll be knowing the extractive value and specific gravity. Specific gravity, as we have seen in physical constants, it helps to maintain batch-to-batch consistency and measure. It is measured using pycnometer or specific gravity bottle. Specific gravity of any given liquid is compared with specific gravity that of water. And these physical constants are mostly constant for each of the material or the source used. Next, in mother tinctures, chromatographic fingerprinting, that is used for quality verification and pH determination. It ensures the stability of tinctures, storage, exposure to temperature and pressure that tend to alter the pH, and in turn make the liquid or the mother tincture unstable. So we need to determine the pH, stability, and specific gravity of the mother tinctures. Next are the standardization of potentized medicines. These potentized medicine, they require special control because they undergo serial dilution and succussion. After, as seen in decimal or centesimal scale, they undergo 1:10 or 1/100th dilution. So you need to find out that one part of the drug present in each of the medicine. For that potentization, Parameters like verification during the manufacturing process, including the dilution ratio, whether you are following 1:10 for decimal, 1:100 for centesimal, and number of succussions you have used, the potentization method followed, equipment validation, what are the advanced measures used or are all to be measured. Next is alcohol strength determination, as in mother tinctures. That is the same for the potentized medicines also, like the percentage of alcohol, purity of alcohol, and its stability is also assessed. Next, coming to toxicology or microbial testing. Total microbial count or total percentage of contamination happening and any sort of pathogenic organisms or decomposition, that is fungal contamination happening to the potentized medicines during transport or during dispensing, that all are to be checked. Then physical parameters like clarity, the appearance, any sort of sedimentation happening to the potentized medicines, and the specific gravity of alcohol or the medicine are to be checked. And container and closure evaluation, they check compatibility of the medicine, sterility, and potentization, protection from light and moisture while transport. As seen, modern analytical methods used in standardization include the HPTLC, nothing but high performance thin layer chromatography fingerprinting, used for identification and determination of compounds from adulteration, determination of any adulteration happened in the given compounds, and perfection and consistency of the given set of drugs that are being tested. Next is HPLC, high performance liquid chromatography, includes the liquid kind of medicines like dilutions, mother tinctures, potencies. They provide quantitative analysis for the marker compounds. Next one is the gas chromatography and mass spectroscopy, useful for volatile constituents that are easily evaporated. Gas spectroscopy is used in compounds that are easily volatile and evaporable. Next is the atomic absorption spectroscopy, used for heavy metal estimation. We are not going in depth regarding this analytical methods. DNA barcoding, modern techniques for botanical authentication of any medicinal plants is also being done these days. And the main source or authoring bodies for standardization are good manufacturing practices, GMP in homeopathy. These guidelines, they are a set of guidelines as mentioned in 11 rules are drafted that are mentioned in Schedule 1 of Drugs and Cosmetics Act 1940, Rules 1945. It is good manufacturing practices are given in that. These guidelines cover the whole manufacturing process from the start of raw material to the premises that are to be covered and personnel, how the person or working in the industry are to be covered, packaging, labeling, how the all these guidelines apply to homeopathic medicines as well as to conventional pharmaceuticals. Failure in applying GMP may lead to major quality and safety concerns such as misidentification, impurity of starting material, cross contamination, or incidental contamination happening. Total 11 rules are given in good manufacturing practices. Here I have covered some 3 or 4. Raw material control, how the raw material are corrected and whether they are authenticated and proper approval is being given for the suppliers, and everything should be documented. Process control, standard operating procedures are to be established. Equipment calibration is being done. Batch records are also maintained. Quality control laboratory, as in ideal pharmaceutical laboratory, that should be maintained, like validated analytical methods and instruments are to be given. Qualified personnel and persons working in the industrial laboratory should follow the rules and they all should be qualified. Documentation systems, everything is documented and monitored. World Health Organization quality systems stress good manufacturing practices and quality control laboratory compliance for pharmaceutical products. And so far, how many research laboratories are there for homeopathy? Firstly, it is Homeopathic Pharmacopoeia Laboratory established in 1975 for the standardization of homeopathic drugs under the Ministry of Health and Family Welfare, Government of India, as the main quality monitoring apex body situated in Ghaziabad. It is approved under Drugs and Cosmetics Act 1940. This laboratory is also recognized by Department of Science and Technology, Government of India. And the second one, Central Council for Research in Homeopathy, CCRH, to carry out all the research activities in different aspects in homeopathy. And next, PCIMH, Pharmacopoeia Commission for Indian Medicine and Homeopathy, Ghaziabad. It acts as an appellate drug testing laboratory for testing and analysis of Ayush drugs. Next is the NIH, National Institute of Homeopathy, Kolkata. Regional Research Institute, Homeopathy, Habsiguda, Hyderabad. Formerly, that has been known as Drug Standardization Unit, operated under CCRH Ayush. And next is the Telangana State Level Drug Testing Laboratory, Kattedan, Hyderabad. State level facility providing analytical and quality control testing services to standardize Ayush drugs. So far, different for Ayurveda, Unani and homeopathy, different laboratories are being found. Regarding CCRH and standardization, CCRH, this council has undertaken pharmacognostical studies on over 368 drugs, physicochemical studies on up to 362, and pharmacological studies on 151 drugs have been studied in all the aspects, covering all the methods of standardization. Standards of homeopathic drugs are covered under second schedule. There's a small technical issue. Yeah, sorry, there was some technical issue. Pharmacopoeias, as for recognized for standardization, Homeopathic Pharmacopoeia of India, HPI, HPUS, Homeopathic Pharmacopoeia of United States, and German Homeopathic Pharmacopoeia. These pharmacopoeias provide standards to test that noted in the required monograms, identification tests, preparation methods, storage requirement, and procedures followed for the testing, both microscopic and macroscopic testing. So far, what are the challenges in standardization of potentized medicines? The drawbacks include extremely high dilution. For 50 millisimal scale or as in centesimal scale, you need to find out for 100th potency, the one part of medicine is to be found. In that kind of highly diluted solution, finding this medicine is challenging these days. But there are advanced techniques, as mentioned, chromatography, spectroscopy, they are detecting the presence of our medicine in the high level as seen in nanoparticle range. That is one of the challenge, and difficulty in quantifying original molecules in high potencies. As I said, in nanoparticle study, the presence of original drug source is to be found. That has become challenging these days. And variation in manufacturing techniques. As said, the preparation may vary from place to place and person to person. Whether even if the advanced machinery are used, the controls and the person handling it may vary. Lack of universally accepted analytical markers for many preparations. At a place, nanoparticle studies are being analyzed so far openly, and there are places where still people are not accepting the homeopathic medicine. So that may be called as a sort of some challenges in standardization. So I'll conclude the session. Standardization of homeopathic drugs and potentized medicines is a comprehensive quality assurance process that begins with authentication of raw materials asset from the start to end and continues through manufacturing, potentization, packaging, storage, quality control testing. And many modern analytical techniques like HPTLC, high performance, thin layer chromatography, liquid chromatography, spectroscopies, also X-ray diffractions have strengthened the scientific quality assessment of homeopathic preparations. The presence of particles or the presence of medicine in high level solutions are all being tested these days. And adherence to pharmacopoeial standards and good manufacturing practice requirements and World Health Organization quality guidelines ensures the consistency, safety, purity, and the reproducibility of homeopathic medicines. For any medicine to be used, we consider whether it is consistent, whether it is safe and purely, and whether it is reproducible as in homeopathic medicines. That's all regarding standardization. Any doubts?
Yes, Dr. Shreya, we have some questions coming up.
Yes, ma'am.
First is how HPLC perform and result the quantity of liquid.
Sorry, ma'am.
How
LC?
HPLC performance result with the quantity of the liquid. So we are basically trying to understand when we can perform HPLC, why do we go for HPTLC?
Regarding liquid chromatography,
Yes.
that's all, ma'am. Regarding liquid chromatography. Am I audible, ma'am?
Yes, yes.
Hello. Through chromatography techniques, we will be able to find out the chemical constituents present in that. And coming to liquid chromatography, the mother teachers are, suppose we need to find out belladonna mother teacher, the physical constituents present in that, like atropine, hyoscyamin, the quantities of that particular phytoconstituents can be known through liquid chromatography.
Okay.
Yes.
And what is the difference with thin layer chromatography? What benefits we get with thin layer chromatography? HPTLC.
Through thin layer chromatography, yeah, HPT in high performance and here, TLC, HPTLC, the main difference is uh, the method that is used. In TLC, maybe single sample or manually it has to be done. The spots are to be used through a capillary tube, and the rate at which we'll call it as a retention factor, the rate at which the solvent is traveled and divided by the component that has been used. So in HPTLC, it is high performance. Nothing but multiple samples can be tested at a time.